Hello, friends of facts, and welcome to 5 Minutes of Science, where we present exciting research from scientific papers. With every video, we will take a look at another inspiring paper from the fields of sustainability, toxicology, chemistry, or climate change. Today's topic is alternatives to animal testing, and we start with a paper by Girand and co-authors published this year in Toxicology Reports. The paper is called Use of LUX, Light Up Cell System, as an alternative live cell method to predict human acute oral toxicity. But before we dive in, let's have a wider look at the relevance of non-animal testing. Every year, approximately 10 million test animals are used in the European Union, 1 million of which with severe impact on their well-being. Most test animals are rodents like rats or mice, and in addition to behavioral research, those tests are mainly conducted for chemical testing. Why is chemical testing important, and do we really need animals for it? Well, we all know that chemicals may be toxic to humans. Just think about alcohol. Others may be even more hazardous. In order to assess their toxic potential and decide whether they shall be authorized or if they require protective measures, they are tested by scientists using laboratory animals. Sometimes, animal tests are conducted even though alternatives are already available. So why is it that animals are still being used in toxicity testing? Due to their complexity, animals are ideal to account for toxic effects, which are often caused by complex mechanisms involving several organs and their interaction. Moreover, the function and structure of organs in mammals, such as rats, are similar to those in humans. But despite these advantages, animal testing is often criticized. Besides obvious ethical concerns, scientists argue that the complexity of animals leads to variable results, which are not reproducible and therefore not meaningful. And despite their similarity to humans, an effect observed in a test animal might not occur or might occur to a lesser extent in humans or vice versa. For those reasons, animal testing shall be reduced, replaced and refined, preferably using alternative animal-free test methods. For example, the EU Cosmetic Regulation prohibits the testing of products on animals since 2013. Today's paper provides an example for an acute toxicity alternative method, which is based on human cells. Acute toxicity is a commonly addressed effect of chemical toxicity. Acute means that a single dose of a chemical causes toxic effects within seconds, hours, or sometimes days. In animal tests, this effect is evaluated by inserting the test chemical into, for example, a rat's stomach per gavage. Then, scientists observe how many of the rats die and at which dose. Girand and co-authors present the so-called LUX method as an alternative to those tests. LUX stands for Light Up Cell System and is used to assess the cell's equilibrium, also called homeostasis. In LUX, human cells are exposed to fluorescent dye. Under normal living conditions, the dye is mostly discharged from the cell with little contact to its DNA. In result, only a low fluorescent signal can be detected. If the cells are exposed to UV light or reactive chemicals, the intake of the fluorescent dye increases, so does its contact to DNA, and hence a strong fluorescent signal can be measured. If now a toxic test substance is added, the homeostasis of the cell is further disturbed, leading to a change in fluorescence compared to a chemical-free control. The degree of this difference in signal is used as a measure for the test chemical's acute toxicity. Summing up, acute effects happen quickly, and LUX is about light coming from cells. A new method like LUX is commonly compared to the standard test method, in this case the animal test. However, the comparison should rather be based on human data. Of course, human toxicity testing is neither allowed nor conducted in the EU, and therefore human data is scarce. How do we have it at all? Well, sometimes humans poison themselves, either accidentally or on purpose. and the resulting effects are recorded while the blood is tested for the responsible chemicals. Such data has been collected by the Acute Tox Project and can now be used for the comparison of lux and human data. Now here's the exciting news. Girond and co-authors could demonstrate that their alternative method is better at predicting acute toxicity levels in humans than the standard animal test in rats. The graph on the right-hand side depicts the correlation of human and lux, the one on the left-hand side the correlation of human and rat acute toxicity. The correlation coefficient R² describes how closely the respective values match. The closer to the line, the better, and likewise the higher R² the better. The conclusion is therefore that lux beats the standard test in rats in the prediction of human acute oral toxicity. Alternative methods to animal testing, like lux, are promising for its replacement. However, it often takes a long time, especially in regulatory toxicology, before they are accepted as standard test method and widely used by scientists. In our next video, we will summarize a research paper that takes a closer look at the obstacles to the replacement of animal testing and how the use of alternative test methods can be improved and accelerated. Have a look at the link in the lower right corner if you're interested. For references and further reading, please see the video's description, which refers back to the encircled numbers in the presentation. Finally, let us know in the comments whether you like this video and which topics we should have a go at in future. And please hit the like button. Thanks for watching and see you soon.